Bismillahirrahmanirrahim. Today we are going to learn about the urine formation by the kidney, how the urine formation take place in the kidney. First of all, we will learn about the steps which are involved in the formation of urine inside the kidney. Actually, the formation of urine take place inside the functional unit of the kidney, that is nephron. You know that nephron is the functional unit of the kidney in which the urine formation take place. The basic steps which are responsible for the formation of urine are four. Number one, glomerular filtration. Number two, tubular reabsorption. Number three, tubular secretion. And number four, water reabsorption. These are the four basic steps which are involved in the formation of urine. We will learn one by one in detail in the next slides. First of all, we should learn about the major role of the kidney and the renal blood flow to the kidney. You know that the blood which comes from the heart through aorta into the renal artery, and then from the renal artery it goes to the kidney. In the kidney, filtration take place. In the kidney, the urine formation take place. In the kidney, the urine, the waste product goes through ureter to the urinary bladder, and the blood which is filtered, which now don't contain any sort of waste material, goes to the renal vein, and from the renal vein it goes to the superior vena cava, inferior vena cava, and then it goes to the heart. And this cycle continues throughout the life and throughout the 24/7 of the human body. Okay, now coming towards the amount of blood flow to the kidney, how much amount of blood goes to the kidney. We can imagine it from the cardiac output. You know that the cardiac output is basically the minute volume. We also call the cardiac output as minute volume, or the cardiac output is also known as the minute volume, because the volume of blood pumped by the heart in 1 minute, that is called cardiac output, or the volume of blood which goes from the heart to the aorta per minute, that is called cardiac output. And we know that each and every minute, or in every minute, 5 liters of blood goes from the heart to the aorta. From the heart to the aorta. Now, what is GFR? GFR is the glomerular filtration rate, and this is also the amount of blood which is filtered by the kidney for unit time, or for minute. For unit time mean for minute. Okay, and we assume the GFR normally in the minute. That is why this is I told that this is per minute. Okay, now how much amount of blood goes to the kidney? Around about 5 liters of blood goes from the heart to the aorta. 5 liters of blood per minute. But this 5 liter of blood, we should focus on this one. For example, per minute, cardiac output, you know that in the cardiac output, per minute, 5 liter of blood goes to the aorta from the heart per minute. Okay, this 5 liter, out of this 5 liter, round about 20% of blood, 20% of blood goes to the kidney. 20% of blood goes to the kidney from the descending aorta. 20% of blood goes from the descending aorta to the kidney, to the renal artery. So what will be the 20% of 5 liter? So 20% of 5 liter will be 1 liter, simple. it will be 1 liter. Okay, we know that 1 liter of blood goes from the heart to the kidney for a unit time, because 20% of the cardiac output reaches the kidney per minute. 20%. 20% mean 1 liter of blood reaches the kidney per minute. Now, out of this 1 liter, you know that this 1 liter blood goes and reach the nephron of the kidney. You know that one out of 1 liter of blood, this whole volume, this 1 liter of blood goes to the goes to each and every nephron of the kidney, reaches each and every combinedly, all the blood is 1 liter. That is for unit time or for minute. Out of this 1 liter of blood, what will be the volume of plasma? So we know that round about out of in the plasma, in the 1 liter, around about say 60%, 60% of blood is plasma, 60% of 55 or 60%, we will assume it at 60%. So 600, we will assume that 600 ml of blood will goes to the 600 of plasma will be there in 1 liter of blood. 600 ml of plasma will be there in 1 liter of blood. That 1 liter which goes into the kidney. Now I'm repeating again this, that in every minute 5 liter of blood goes to the aorta from the heart, and this 5 liter, 1 liter of blood reaches the kidney. That is 20% of blood reaches the kidney per minute. So 1 liter of blood reaches the kidney per minute. So out of this 1 liter, this 1 liter is composed of 600 ml of plasma and 400 ml of cell material, or blood cells and protein, etc. But 600 ml of this contain plasma. 600 ml of plasma is there in the 1 liter of blood. So this 600 ml, this 600 ml of blood reaches which portion? This 600 ml of blood, this 600 ml of plasma actually reaches the glomerulus of the nephron. Glomerulus of the nephron. This 600 ml of plasma reaches, you can say that 1 liter of blood reaches this glomerulus, but out of this 1 liter of blood, we will assume that 600 ml of plasma reaches this glomerulus, and this 600 ml of blood is filtered in this glomerulus. This is the magnified shape of this glomerulus. This is renal corpuscle. Renal corpuscle is the combination of Bowman capsule and glomerulus. So 600 ml of plasma reaches this glomerulus each minute. In each minute, 600 ml of blood reaches, 600 ml of plasma reaches this glomerulus. Okay, now this 600 ml of plasma is filtered in each minute through the Bowman capsule. So out of this 600 ml, 120 ml of filtrate reaches the Bowman capsule. Out of this 600 ml, 120 ml of plasma or 120 ml of filtrate reaches the Bowman capsule. 600 ml. 600 ml reaches the glomerulus. While out of this 600 ml, 120 ml of filtrate reaches the Bowman capsule. From 600 ml of blood plasma, which is present in the glomerulus, 120 ml of filtrate reaches the Bowman's capsule. Okay. Now I think this is almost clear to you people. uh, okay, so you people learned now that in every minute 120 ml of blood reaches which portion? The Bowman capsule. 120 ml of filtrate reaches the Bowman capsule. This is called glomerular filtration rate, GFR. This is what we call GFR, which I have told in the previous slide, that GFR is the glomerular filtration rate. Glomerular filtration rate is the if someone asks you that what is the value of glomerular filtration rate, so in the normal adult, this is 120 ml. Glomerular filtration rate is the rate of filtration of blood in the glomerulus, or the rate of filtration of or the filtration of blood in the renal corpuscle per minute. That is glomerular filtration rate, and that is 120 ml. So out of 5 liter of cardiac output, out of 5 liter of cardiac output, 120 ml of filtrate reaches the Bowman capsule. Now this is the Bowman capsule. Uh, how this reaches this Bowman capsule? Actually, the first step of the urine formation is the glomerular filtration. So, in the glomerular filtration, you should learn about that how this filtration take place. The filtration take place in this way that this is the afferent arteriole, and this is the efferent arteriole. This is the afferent arteriole, and this is the efferent arteriole, and this is the glomerulus. And the glomerulus is highly perforated, and you can see over here that this glomerulus is highly perforated. This perforation, it means that glomerulus contain large number of pores. So, but if there are pores are already over here and over here also. But why this glomerular filtration? Why this filtration take place? Why this blood or this plasma leak out from the glomerulus to the Bowman capsule over here? Why not it leak out to over here? Because these endothelial cells also contain pores. Why this glomerular filtration take place over here? Why this filtration? Why this blood is filtered over here? Why not it is filtered over here? Because uh, this contain perforation, but this also contain perforation is also over here. But the only difference which you should learn about, that this is the afferent arteriole through which the blood goes to the glomerulus. This is the afferent arteriole through which the blood goes into glomerulus, and this is the afferent arteriole over here. This is the afferent arteriole, this is the afferent arteriole. Through afferent arteriole, the blood goes to the glomerulus. The diameter of the afferent arteriole is very large. You can see over here that the diameter of afferent arteriole is very large, while this is the efferent arteriole through which the blood goes from the glomerulus to the efferent arteriole. And this is the this is the outgoing blood vessel, and this is the upcoming this is the incoming blood vessel, and it is the outgoing blood vessel. So in the incoming blood vessel is having large diameter, while the outgoing blood vessel is having small diameter. If you connect two pipes with each other and with a junction, and that junction will be the glomerulus, and if that junction is a bit perforated, if that junction is a bit perforated, and you join two pipe, the joining pipe is smaller in diameter. If the joining pipe is smaller in diameter, and you apply water pressure in the in the big diameter, in the pipe which is having large diameter, what will happen? This blood, if goes, this amount of blood which reaches over here, 600 ml of blood reaches over here each minute. 600 ml of blood reaches over here. 600 ml of blood reaches over here. But when this 600 ml blood reaches over here, this blood vessel is not having that much capacity to to allow 600 ml of blood, because the diameter of this blood vessel is very small. This blood vessel is having small diameter. This do not allow 600 ml of plasma per minute, and this can allow 600 ml of blood per minute. So what will happen? The only difference will be the filtration. This difference will be compensated by the filtration, and that filtration is called glomerular filtration. So you have learned over here that the efferent arteriole is having high diameter, large diameter, and the efferent arteriole is having small diameter. So due to this difference in diameter, the filtration takes place. The filtration takes place over here. Okay, this filtrate this filtrate contain large amount of glucose. This filtrate contain large. This filtrate do not contain any sort of blood vessel. In normal physiology, this filtrate do not contain any sort of blood vessel. This may contain some debris. This may contain some proteins also, but this do not contain any blood cell. So this contain large amount of glucose. This contain large amount of sodium, chloride, calcium, etc. But this do not contain any sort of blood cell. So this blood reaches over here. So this is what we call glomerular filtration. And this glomerular filtration, why this glomerular filtration take place over here? Because of the difference in diameter of the efferent and efferent arteriole. So from the Bowman capsule, this goes to the proximal convoluted tubule of the nephron. This was all about the glomerular filtration, and the glomerular filtration rate, the normal glomerular filtration rate is 120 ml. 120 ml mean 120 ml of filtrate reaches from the glomerulus to the Bowman capsule per minute. Now coming toward the tubular reabsorption. This is the second step of the urine formation. In this very step, what will happen when the filtrate reaches the proximal convoluted tubule? This is the proximal convoluted tubule. In the proximal convoluted tubule, you should imagine that the surrounding cell, the surrounding blood vessel, these blood vessels go and surround these convoluted tubule. This efferent arteriole goes around these are intermingled around the proximal convoluted tubule and distal convoluted tubule, and also around this area. This is the Bowman capsule of the nephron, or descending and ascending tubule. Okay. So this in the proximal convoluted tubule, what will happen in the proximal convoluted tubule? The glucose will be absorbed. The sodium and glucose will be absorbed to these blood vessels. The sodium and glucose will be reabsorbed. This is called tubular reabsorption. And tubular reabsorption is the step in which the water or the solute goes from the tubule to the capillaries, or the transfer of various material, including the water, from the convoluted tubule or from the tubule into the blood vessel inside the nephron. That is called tubular reabsorption. And this reabsorption takes place in the proximal convoluted tubule. This reabsorption takes place in the descending loop of Henle. This also takes place in the ascending loop of Henle. This also takes place in the ascending tubule. This also takes place in the distal convoluted tubule also. So this is called tubular reabsorption. Tubular reabsorption is the absorption of various materials, absorption of various solvents and solutes from the nephron to the bloodstream. This is called tubular reabsorption. And in this area, the salts and glucose will be reabsorbed. Sodium and glucose will be reabsorbed over here. But as it goes downward, there is a small logic, but which work very efficiently over here. That why the reabsorption takes place over here, why not, and why this nephron goes downward and this then upward. Why not this goes uh, just in the same manner over here and goes to the collecting duct? Why not? Why this is having a ascending and descending tubule? Why this is having loop of Henle? And there is a small logic which work efficiently. You should learn it right now. That when the this area of the normally this area of the nephron is in the above this area. This is in the cortex of the mid of the kidney, while this area below this line, below this line, this area is inside the medulla of the kidney. So what will happen? In the cortical region, the osmotic pressure around this convoluted tubule, the osmotic pressure around this convoluted tubule is very low, while in this area, as it goes downward, the osmotic pressure around this convoluted tubule goes very high, goes very high. So you can say that the osmotic pressure, if the osmotic pressure in the proximal convoluted tubule over here is 300, so the osmotic pressure over here will be 1200. There is a big difference. There is a big difference. As it goes downward, the osmotic pressure around this convoluted tubule goes high, and as it goes upward, the osmotic pressure around this convoluted tubule goes downward. The osmotic pressure decreases as it goes upward, and the osmotic pressure increases around this tubule as it goes downward. So what will happen when the osmotic pressure decreases? Osmotic pressure means that the if we have two solution, one solution is having 200 milligram of sugar, one solution is having 200 milligram of glucose per liter, and the other solution and the other is having 20 milligram of glucose per liter. One is having 200 milligram per liter, and the other is having, for example, I am writing it over here that we have two solution. One is having 200 milligram of sugar per liter, and the other is having 20 milligram of sugar per liter. So the osmotic pressure of this will be very high. This will have high osmolarity. The osmolarity of this solution will be very high. The osmotic pressure of this solution will be very high. The osmotic pressure of this solution will be very low. So if the osmotic pressure, if we give a layer in between these two solutions, so what will happen? The solvent will goes from the lower concentration to the higher concentration. And you should also learn that this area is not permeable to this area from the ascending from the proximal convoluted tubule to loop of Henle. This area is not permeable to from this area, not from this area, from this descending tubule, and this loop of Henle is not permeable to any solute. This is permeable to water. This is highly permeable to water and sparingly is permeable to solute. If this was permeable to solute, then there will be no problem. But this is not permeable to solute. This is highly permeable to solvent. This is highly permeable to water. So what will happen? It will eventually, the inside the osmotic pressure inside the this convoluted tubule, inside the filtrate, then the convoluted tubule will not be more than 300, while the osmotic pressure around this convoluted tubule goes high, it goes downward. So if it goes downward, the water will be leak out from the tubule into the external environment, and this water will be absorbed to the capillaries, or to absorb to the capillaries and these blood vessels. So what will happen as it goes downward, the water reabsorption take place. The water reabsorption take place because of difference of osmotic pressure, because of difference of osmolarity. As the osmotic pressure become high, the filtration, the reabsorption become high. As the osmotic pressure become low, the reabsorption become low. It depends upon the osmotic pressure. So when it reaches over here, there is no round about 90% of water is reabsorbed from this from this filtrate, because of the high osmotic pressure around, because of the high concentration of the solute in the cells around this loop of Henle. And this water will be absorbed into the capillaries. So this will goes upward, and as it goes upward, this area, this ascending loop up, this ascending convoluted tubule, this ascending tubule is permeable to solute. Now this is permeable to solute, and sparingly permeable to water, not highly permeable to water, and sparingly permeable to water and highly permeable to solute. While this area is highly permeable to water and sparingly permeable to solute, while this area is highly permeable to solute and sparingly permeable to water. So what will happen when it goes upward? You have already learned that over here, the solution inside the filtrate inside the tubule is highly concentrated. The filtrate inside this tubule will be highly concentrated. Why this will be highly concentrated? Because of the compensation of the osmotic pressure, because of the transfer of the water from this tubule into the surrounding, because of the transfer of water from the tubule into the capillaries. So when it goes upward, when it reaches over here, there is very small amount of water over here in this tubule. So what will happen? As this area is impermeable to water, so the water cannot cross, the water cannot come from the surrounding to this tubule. So what will happen for the compensation of the osmotic pressure? What will happen? This solute will be transferred from this area to the surrounding. The solute will be transferred from this area to the surrounding. Which type of solute? For example, the calcium and the potassium will be reabsorbed from this area. The calcium and the potassium, especially not the sodium, the calcium and the potassium will be reabsorbed from ascending tubule to the surrounding and to the capillary. So when it goes over here, then the water is also reabsorbed from this area. This area is now permeable to water, so some of water is reabsorbed over here, because this once again this area is permeable to water, so the water will be reabsorbed. So after this tubular reabsorption, what will happen? The next step is tubular secretion. The next step is tubular secretion. Tubular secretion is the secretion of secretion into the tubule, secretion of materials from the blood stream to the tubule, from the blood stream to the tubules. Secretion of materials, secretion of anything, the secretion of solute and solvent from the blood into the tubule. This is opposite to the tubular reabsorption. In the tubular reabsorption, the solvent and solute will reabsorb from the tubule to the bloodstream, while in the tubular secretion, this will be reabsorbed from this will be transferred from the blood stream to the tubule. So the reabsorption, the tubular secretion mainly take place in the distal convoluted tubule. In the distal convoluted tubule, the tubular secretion take place. Uh, this this is secretion. This arrow represents the secretion. So the tubular secretion take place over here. Now, so some of the you can say is some of the sodium and water and urea is secreted from the surrounding capillaries into the tubule over here in the tubular secretion. In this step, uh, the water, sodium, and uric acid, etc., are secreted from the surrounding capillaries to the from the surrounding blood vessel to the tubule, and that mainly take place in the distal convoluted tubule. This is called the tubular secretion step. And this in the tubular secretion, large amount of water is also secreted from the large amount of water is secreted from the capillaries or the blood vessel to the tubule. Why the blood the why this tubular secretion take place? This also take place due to difference in the osmolality, because while reaching over here while reaching over here in this area, the osmotic pressure in the surrounding become low and the osmotic pressure in this area is high. So to compensate the osmotic pressure, to make the osmotic pressure isotonic, to make it the solution outside the tubule and inside the tubule isotonic, so this this tubule will receive the large amount of water in this area. Now coming towards the next step. Before coming toward the next step, you should focus on this one. The this There are two types of nephron inside the inside the kidney. One is the cortical neuron and the other is the juxta-medullary nephron. Cortical nephron and juxta-medullary nephron. This one is cortical nephron. The large portion of cortical nephron is in the cortex region of the kidney, and the smaller portion of this, especially the descending tubule and the loop of Henle, is inside the medulla of the kidney, while the juxta-medullary nephron contain large portion of this is inside the medulla, and small portion of this is inside the cortex of the kidney. So this comprise of 15% of the nephron, and this comprise of 85% nephron. 85% nephron inside the kidney are cortical neuron, while 15% of nephron inside the kidney are juxta-medullary nephron. And these nephron produce concentrated urine, and these nephron produce dilute urine. Why? Because of the difference in the osmotic pressure, as I have already discussed in the previous slide. Now coming toward the next type, water reabsorption. This is the very important step of the last step and very important step of the urine formation. What will happen? The reabsorption takes place in the collecting duct, and you should learn that the collecting duct is not the part of single nephron. Collecting duct is the part of multiple nephron, not only one nephron. Large number of nephron combined together, and the distal convoluted tubule goes into the collecting duct. One collecting duct, large number of distal convoluted tubule goes into the one collecting duct, and that is that receive uh, the filtrate, the final filtrate, which become urine in the after passing through the collecting duct. After passing through collecting duct, it become urine. It will be no more filtrate. This will be called urine onward. So when it reaches this collecting duct, the reabsorption takes place. Reabsorption is the step in which the water and solute are reabsorbed from the collecting duct to the surrounding, are reabsorbed from the collecting duct to the bloodstream. To surrounding mean to the bloodstream, and because it will eventually go to the bloodstream from the surrounding. Okay, what will happen when when the filtrate reaches the collecting duct? In the collecting duct, inside the collecting duct, what will happen? There is sodium potassium channel. There is sodium potassium channel, and this sodium potassium channel, this absorbs sodium and secrete the potassium. This exchange sodium with potassium. This absorb in the distal convoluted tubule. The sodium is reabsorbed, and potassium is secreted from the surrounding capillaries, from the surrounding blood vessels. The sodium is the potassium is secreted, and the sodium is reabsorbed through sodium potassium channel. This takes place simply through diffusion. This the chlorine is also reabsorbed, and this why this potassium is secreted, because large amount of potassium is reabsorbed in the distal convoluted tubule. So to compensate the volume of the amount of potassium inside the body, so this potassium will be reabsorbed, will be secreted, resecreted over here, uh, with each sodium, with the reabsorption of sodium, the potassium will be secreted. If the potassium is not secreted, there will be no reabsorption of sodium. There will be no absorption of sodium. The sodium will be absorbed only and only by the secretion of potassium. So the this step was actually the water reabsorption. So with the absorption of sodium, the water molecule will be also reabsorbed, because each sodium will have some molecule of water all around. So the water will be also absorbed over here with the sodium, but simple water, which is not combined with any ion of sodium, is also reabsorbed over here in this step. And that cause the osmoregulation of the body. So this step contains the reabsorption of water. This step comprises of reabsorption of water from the collecting duct. And this reabsorption, this is actually there are two types of absorption uh, inside the body, inside the nephron. This absorption is called facultative reabsorption. I will talk about this facultative reabsorption in the next lecture, but you should learn that this absorption can be modified. This absorption can be modified absorption. This absorption of water uh, change with the time and with the condition of the body, with the condition of environment and the condition of the body. This depend upon the secretion of ADH. ADH is a hormone that is called antidiuretic hormone. That is called antidiuretic hormone. ADH, antidiuretic hormone. So this antidiuretic mean antidiuresis. Antidiuresis mean urine, anti-urination or anti-maturation. So this hormone is against the volume of the against the high volume of the urine. Mean this is antidiuresis. This is against urination. This hormone is against urination. Why this is against urination? Because this volume of blood, this hormone actually decreases the volume of urine over here. This decreases the volume of urine. How this decreases the volume of urine? How this decreases the volume of urine? This decreases the volume of urine in the manner by reabsorption of water. If the water is reabsorbed from this collecting duct, so the volume of urine will be decreased. If this antidiuretic hormone is secreted in high amount, very low amount of urine will be secreted over here. If this antidiuretic hormone is secreted in small amount, then very small amount of urine will be excreted through the collecting duct. So when the amount of ADH is high inside the body, the urine formation will be low. If the amount of ADH is low in the body, the volume of the urine will be high. And if there is no secretion of ADH, then that is called diabetes insipidus. Diabetes insipidus. And diabetes insipidus is the condition in which there is no secretion of ADH in the body. One is diabetes mellitus and the other is diabetes insipidus. In the diabetes mellitus, the secretion of There is no secretion of insulin and diabetes insipidus. Insipidus. There is no secretion of ADH, and if there is no secretion of ADH, there will be high amount of secretion of excretion of urine. There will be high amount of formation of urine inside the body. So the antidiuretic hormone actually make uh, aquaporin channel. This hormone synthesize aquaporin channel, which is responsible for the reabsorption of water. These for the reabsorption of water will take place over here. This is actually the reabsorption of sodium and sparing of potassium, while the reabsorption of sodium also take place in the distal convoluted tubule, and which take place, that is called facultative reabsorption, and take place inside this due to the secretion of due to secretion of ADH. If there is no secretion of ADH, there will be no secretion, there will be no reabsorption of water. So in the whole lecture you have learned about that 120 ml of plasma reaches over here, 120 ml of filtrate reaches over here out of 600 ml of plasma inside the Bowman capsule. And when normally, not per unit time, normally per unit time 120 ml of urine reaches over here, 120 ml of filtrate reaches over here, and this, out of this 120 ml of filtrate, how much amount will be transferred to the urinary bladder or to ureter, or how much amount will be transferred into the collecting duct into the pelvis or to the ureter. So you should be clear over here that per minute only 1 ml of blood goes from this area to this area, only 1 ml out of 120 ml of out of 120 ml of what? Out of 120 ml of filtrate which reaches the Bowman capsule, only 1 ml of blood in the normal condition. In the normal condition, 1 ml of urine will reach to the ureter. 1 ml of urine will reach to the ureter per minute. So in 1 hour, 60 ml of urine formation will take place. 60 ml of urine formation will take place. So the rate of urine formation is 60 ml per minute. 60 ml per minute. 60 not 60 ml, 1 ml per minute. 1 ml per minute. The GFR is 120 ml per minute. The GFR is 120 per minute, while the urine formation is 1 ml per minute, and that is 60 ml per hour. So this was all about. This was all about the information of urine, but you should learn that in the 1 hour, only 60 ml of urine goes to the urinary bladder. Only 60 ml of urine goes into the urinary bladder, and this urinary bladder can compensate up to half liter of blood, up to half liter of urine.